Okay, welcome to the session on analysis. I'm Yolanda Gil. I'll be sharing the session. And what I'm going to do is we have 10 minutes per speaker. I'm going to do like this, signal to them when there's 2 minutes left. And then I'm going to go like this when they need to cut off. Hopefully we can stay on track. And we have time at the end for questions, the same format as yesterday. And our first speaker is James Sluka.
Good morning, everyone. I've changed my topic from what's in the handout. There's a new topic there. And if I can get, there we go. So, modelers have a workflow. We don't necessarily know what that term means. Different modelers have different ways of doing workflows, but this is one kind of typical way that you might actually go about generating a biological model. Starting with some biological observation or some biological system that you're interested in modeling, create a biomodel where you describe the biological components that you're going to have, create a mathematical model, for instance, it's a set of ordinary differential equations. You then create a computational model, which in some way instantiates that set of ODEs. You then finally get to the point where you get to run a simulation. Hopefully you get as a result, hopefully you make some prediction of that, from that result, and then there's some cycle back up to the top to redo the whole process. In practice, you tend to jump back up from almost all of these intermediate stages back up to the top. You find out that you did some biology you don't know, and you have to go back to the biologist, get some more information. So there's a lot of iterative loops in this process. People tend to do something like this when they develop a model. Unfortunately, when they publish a model, this tends to be what actually gets into the paper. The biological observation or the biological challenge they're studying and the results of their simulation. And all of this intermediate work kind of vanishes. Some of it's in there in the paper, some of it's not. Some of it the modeler is aware they did, but it's not codified or recorded anywhere. And often modelers don't even make a prediction. They just publish the result and let somebody else figure out what it means. We're losing a lot of data. This is kind of a typical publication you might see. This is a multi-scale model of tumor growth with angiogenesis. It has small molecule diffusible fields. It has growing cells that divide and multiply. It has vasculature growing, supplying oxygen. Typical multi-scale, multi-cell based model. In the publication for this paper, most of the biology described in this one table. It says that there's a cell type called normal, and it proliferates, and it consumes oxygen, and it changes to hypoxic, and it changes to necrotic, and there's a cell type called hypoxic, and there's vascular cells. Vascular cells supply oxygen at particular tension. That's basically the description of the model, and it's in a table in the paper. The paper also has some screenshots of the simulation running. Somewhere else, perhaps in a supplement to the paper, is the source code that generated that model. That source code is not linked to this table in any logical way. So where is normal cell in the source code? What's it called in the source code? So we don't really have a good link between what computation was run and what the biologist or what the modeler was trying to simulate. Furthermore, this description is not machine searchable. This description is probably really not machine searchable either. Not only is this possibly a text with the author-chosen wording, sometimes this is a figure. It's a bitmap. So there aren't even any words. It's just a bitmap image. Beyond that kind of problem, we have this wonderful tool called Google. It's probably the largest computing resource in the world that's freely accessible. Google has certain requirements, though, on the kinds of documents it can index. And interestingly, OWL, the web ontology language, ontologists like to misname things. Here's a little snippet of OWL. If Google sees this snippet of OWL, which is an RDF XML, the only thing that Google indexed out of all this is an ontology about pizzas, which is kind of boring, but it's about pizzas, is this one number, 900. Out of all that XML, Google sees the word 900. This XML is incorrectly done to be indexed by Google. All kinds of other file types are indexed, Word, PDF, plain text, source code, all kinds of things are indexed by Google. This vanilla XML is not indexed by Google. We can annotate this stuff. There are ways of doing that. One kind of off to the side a little bit, the whole idea of the conference is we're really talking about cross-scale models. We're going to have models at multiple scales. We really should have some best practices. Our personal belief at the IU group is that the models at every scale should be modeled using the standard model at that scale. Our standard model for ODE-based reaction kinetics. A user should not be writing a MATLAB application to solve their ODEs. There are standard models at that specification. Whole body modeling, PBPK, is a standard for how you describe PBPK models. You shouldn't be writing one of code to your particular whole body model. We don't have a lot of work yet. Ultimately, we probably need some standard API interfaces. Whatever you're doing needs to be biologically annotated. You need to describe the biology you're modeling. This is a very long-term one, but my personal view is any multi-scale model, the model at every scale should run on itself by itself. If you've got a PBPK model and a subcellular reaction kinetic model, you should be able to run those two models completely separately. You should be able to validate and develop and annotate each one of those models separately before you put them together. Another enabling technology is that we have a lot of data, and it's in a lot of different formats. Here's a Somitogenesis example where you have image data, you have microarray data, and you have a computational model. James Glazer actually showed this model very briefly the other day. All of these models, these are all models. This is a model. This is just as much a model as this model. But this image data, this data, and this data should all be annotated in a common way. In particular, they should all have the same biological descriptors associated with it. This particular sets of data are in chicken, Somitogenesis, and it's hour, or whatever it is, 12 hours post-fertilization. That common biological description describes the biology for each of these models. That common biological description allows me to find a computational model, a microarray, an image stack, epidemiological study, whatever you want. It's that common biological description that lets us find all that. Each one of these files would also have its own specific metadata. This is taken on a Nikon microscope, whatever illumination. This was done on an Affymetrix chip. This simulation was done in Composite. Whatever specific metadata is associated with that particular modeling scale. There's really no reason why we can't do this now. We're not doing it now, though. We believe the biological description should be directly embedded or linked to the file that's actually executed. Your source code should have the biological descriptors associated with it in some way. You should also describe ontologically, or using a controlled vocabulary, the actual computational techniques you used. What was that ODE solver that you pulled off the shelf to solve your ODEs? You should name that ODE solver. The relations between the biology and the computation should be described. My reaction kinetic network is solved in this ODE solver. The results that your code produced should be annotated, and the results should be linked to the source code that created it. One approach to how we might do this. We have two kinds of people in the world. We have people that create data, and we have people that consume data. These people are often the same people. Beginning of a project, I tend to be a data consumer. Late in a project, I tend to be a data creator. How can we get these people creating and searching for data in an efficient manner? One way we do that is get them to agree to use the same reference ontologies. Use the bio-ontologies like foundational model anatomy, KEBI, gene ontology. Agree to use those as our keyword lists. To create a data source, our data creator goes to some simple web-based tool, enters his image, enters his source code, enters his picture, enters whatever his data is, and says, I want to annotate my process of mitosis. What's the correct ontological term for mitosis? Goes out, gets that term, adds it to the file, publishes the file somewhere. Maybe it goes into a repository, maybe it goes into just a web server somewhere. Data searchers, I want mitosis. What's the correct term in the ontologies for mitosis? Okay, I get the correct term for mitosis. I now use Google to search all of these files, no matter where they're at. I don't need to know what database they're in. I can find the data. I can at least find the link to the file that contains the data. This is a demo that we did last summer. This is actually a Python script a little tool that lets you highlight words in the Python script. One word is APAP, which is an abbreviation for acetaminophen. And this red is Python comments that have been added, including the KEBI for acetaminophen, its alternate names, paracetamol, all directly embedded into the source Python code. This Python code is searchable by Google, and if somebody queries for acetaminophen, they will find this file. If they do a more complex search, they will only find this file. In fact, if they include this distributed biodata biomodel resource annotation, they will only find this file if they query acetaminophen. And I think I'll stop there because I think I'm on time. Thank you.